Hello all, I am Dr. Atanu Roy, Senior Resident from Department of Physiology, AIIMS. This is paper number 8, Medical Physics, and module number 4, which is the basics of electromyography. The objectives of this module are the introduction, the physiology of muscle activation and force generation, the biophysics of generation of electromyographic signals, and the electromyography instrument and its various components. We will discuss then the recording of the signals, and finally we will conclude it with summary. Here we have seen that the integration of physics with medicine has opened the doors for research and diagnostic tools in understanding the pathophysiology of disease. One such example is the electromyography. In this module, we will discuss more about electromyography. Electromyography is the study of the measurement of the electrical activity of the muscle. Electromyograph is the instrument used for recording the muscle activity, and electromyogram is the record or the tracing made by the electromyograph. Electromyography plays an very important role in our understanding of the muscle activity, but to understand the principles of electromyography, we must know how a muscle is activated, a force generated, and signals recorded. In this module, we will be talking EMG as a short form for electromyography. Now, let us see the physiology of muscle activation and force generation. The concept of motor unit is that the motor unit consists of a motor neuron and the muscle fibers innervated by the axon. It is the functional unit of the neuromuscular system. Let us understand each component of motor unit separately. The group of motor neurons that innervate a single muscle is called motor nucleus or the motor neuron pool. There are two types of motor neurons, the alpha motor neurons and the gamma motor neurons. Both the classes of the motor neurons are unique in their ways. The alpha motor neurons are like the alpha males, big and innervate lots of extrafusal muscle fibers, whereas the gamma motor units or the gamma motor neurons are smaller than the alpha motor neurons, which innervates mainly the intrafusal muscle fibers. From the motor neuron pool, all these neurons come out from the spinal column of spinal cord or the ventral column of the spinal cord. So, when an action potential reaches the motor neuron, it passes down to the different muscle fibers and activates them as well. The question is, how does the muscle get or receive these action potentials? It is receiving like a single wire without any breaks in between, or is there any discontinuity between them? To understand this, let us learn about the neuromuscular junction and how the action potential passes through them. In a motor unit, the nerve synapses with the muscle fiber. The terminal end of the neuron is the presynaptic part. Then comes the synaptic cleft, which ends with the postsynaptic membrane of the muscle fiber. The terminal end of the nerve, otherwise the presynaptic membrane, have various spatial densities which are called as the synaptic button. These specialized locations mainly consist of neurotransmitters. The action potential when arrive at the presynaptic membrane, these neurotransmitters are released and they diffuse through the synaptic cleft and attaches with the postsynaptic membrane, thereby producing end plate potentials. These excitatory end plate potentials summate further and develop the action potential. This action potential then brings out the muscle contraction. Let us understand the anatomy and contraction of the skeletal muscle. Understanding muscle structure. The muscle bundles consists of large number of muscle fibers. Each muscle fiber is equivalent to a cell. Each fiber is made of myofibrils that in turn consist of densely packed contractile components which are arranged in a regular fashion. It gives a striped appearance under the light microscope. Each myofibril consists of a thick filament, otherwise myosin, and a thin filament, that is actin. It is the interaction between these two filaments which brings out the contraction of the muscle in a coordinated fashion. Now, the figure 1 shows the diagram. The skeletal muscles, they are the most abundant and are attached to the skeleton. That is why they are called as the skeletal muscles. They are also known as striped, striated, somatic and voluntary muscles. They are innervated by somatic nervous system, so they are under voluntary control. So, we are able to move the muscles at our will. They are also quick to respond to stimuli and are capable of rapid contraction and get fatigued easily because of their rapidity. Each muscle fiber is cylindrical, multinucleated cell containing groups of myofibrils. The myofibrils are in turn made up of myofilaments, that is of 3 types: actin, myosin and tropomyosin. Examples of the skeletal muscle include all muscles of the body wall. Now, if we see a muscle under the microscope, here we find that under light microscope there are darkly stained areas called as the A band and lightly stained areas called as the I band. The main composition of A band is mainly the myosin and I band the actin. The myofibril consists of repeated patterns of lines and bands. Between the two Z lines, the portion of the myofibril is called as sarcomere. It is the contractile unit of the muscle. I band lies immediately adjacent to the Z line. The A band lies in the center of the sarcomere and H band is situated in the middle of A band. Now, let us see about the sarcotubular system. The endoplasmic reticulum is modified to form sarcoplasmic reticulum. It in turn forms a dense network around the myofibrils. It helps to distinguish between the myofibrils from the myofilaments. The cell membrane around the muscle fibers invaginates transversely deep inside, helping in spread of wave of excitation throughout the muscle. This is called as the T-tubules. At the AI junctions, the sarcoplasmic reticulum comes very close to the T-tubules. These are the areas where the tubules of sarcoplasmic reticulum are enlarged, forming a cisternae, which is a sac-like structure. When the two cisternae comes close to a T-tubule, it forms a triad. The concept of excitation and contraction coupling is very important to be understood. Now, let us see. When an action potential is generated, it traverses along the sarcolemma, and as we have studied earlier that the sarcolemma invaginates deep inside the muscle fiber in the form of T-tubule. So, when the action potential is generated, it dips down deep inside via the T-tubules. It in turn helps to spread the wave of excitation deeper inside the muscle fiber. The sarcoplasmic reticulum comes closer to the T-tubules at the AI junction, thereby forming a triad, which the action potential reaches the T-tubule. It opens the DHPR receptors, that is dihydrogen pyridine receptor. The DHPR is voltage sensitive. They undergo a conformational change, which in turn leads to the release of calcium ions from the ryanodine receptors, which are located on the cisternae by physical interaction. The ryanodine receptors are calcium channels that help in release of calcium from the cisternae. The DHPR and the diodine receptor interaction releases calcium from the cisternae. thereby increasing the concentration of calcium inside the sarcoplasm of muscle cell, which in turn help in contraction of muscle. The relaxation of the muscle depends on the active transport of calcium back into the sarcoplasmic reticulum. Let us discuss the biophysics of the generation of the EMG signals. By now, we have understood the basics of anatomy of the skeletal muscle and how a muscle gets activated and contracts. In this section, we will try to understand the generation of action potential, how the EMG signals are generated, and what is the concept of crosstalk while recording EMG. Let us come to the first question, that is the generation of the action potential. In experiments involving insertion of a micropipette electrode inside the muscle fiber, it is found that the cell is negative inside with respect to the extracellular environment outside. The resting membrane potential of 70 to 90 millivolt is mainly maintained by the presence of the sodium potassium pump that continuously works against the concentration gradient of ions flowing through the membrane. With the arrival of action potential at the presynaptic membrane, acetylcholine is released that passes through the synaptic cleft and excites the postsynaptic muscle fiber membrane, producing a local potential gradient. This excitation is called as depolarization, and it is during this phase when there is rapid inflow of positive ions that renders the inside of the cell positive. The outside of the membrane is negatively charged, so positively charged ions from the neighboring areas will get attracted towards it, as it is depicted in figure number 2. This process continues and the propagation of action potential in this manner is called electrotonic spread. Theoretically speaking, this spread can occur in any direction. This holds good in the case of muscle, but in nerves, the electrotonic propagation is unidirectional because of unidirectional transmission in neuromuscular junctions and synapse. Every wave of depolarization is followed by repolarization, and this process repeats itself over and over again till it reaches the tendon endings. We know that a current flow is also associated with potential changes. This propagating action potential creates a transmembrane current profile that also travels along the membrane of the muscle or sarcolemma. For the muscle fibers, the length of the depolarization and repolarization are very small, otherwise in millimeters. So, we can consider a muscle fiber a very thin tube in which the current flows only axially. In other words, it can be considered as a line source model. Figure 3 in the line diagram is the line diagram of the linear core conductor model, where Z is assumed to be the center of the fiber, the subscript small I represents the intracellular and subscript E represents the extracellular. R represents the resistance, I represents the capital I represents current, and E represents the extracellular potential, and small I represents the intracellular potential. Now, the decrease in potential per unit time is equal to the product of resistance per unit length and current passing through it. Now, in this following equations as given in the module, it is seen that the intracellular is given by equation and extracellular is given by the later equations. The motor unit consists of motor neurons and the muscle fibers. When an action potential is generated in the neuron, it activates the muscle fibers associated with it. The degree of depolarization of different fibers varies based on the distance of each fiber from the source. The summation of all this potential is called as the motor unit potential. The electrodes record this potential and depict it as the EMG on the computer interface. Let's understand the concept of volume conductor. The biological tissues separating the sources and the recording electrodes are called as the volume conductor. It plays a very vital role in the acquisition of signals. The generated action potential creates an electric field in the surrounding space. Thus, the potential generated by the motor unit can be detected by a or at a distant location. The potential signals have to traverse through various media so as to reach the recording electrodes. Under static conditions, the volume conductor obeys some relations which are given in the A text, where the J is the current density in the volume conductor, capital I is the current density in the source, and the sigma represents the current density in the source. Now, what happens if the media is homogeneous? The conductivity does not depend on the site of recording, but if the media is non-homogeneous, then the conductivities across various media and distance of the electrode and source play a very crucial role. Now, how do we understand about these things? Before we go further, we have to understand the concept of crosstalk. Crosstalk is the signal that is detected over a particular muscle, but it is actually generated by a nearby muscle. It is the far field potentials that are being generated by the dying of the potentials at the tendons. This phenomena is mainly seen in surface recording. The properties of volume conduction comes into play when the far field signals travel along the surface and tissues. The main problem with the phenomena of crosstalk is that they influence the interpretation of the EMG recordings. Some important concepts of crosstalk that we must remember is that it is mainly by the far field signals that are the results of the extensions of the potential at the tendons. Then, when we talk about the shape of the signals over the active muscle, the crosstalk signals are very different from the signals of the active muscles. The bandwidth signals of the crosstalk is larger than the signals from the active muscle. The component of the EMG instrument is very unique to be understood. Now, in this next segment, we will be trying to understand the various components of the EMG equipment. Now that we have understood the genesis and progression of action potential, let us take our discussion a little further. In this segment, we are trying to answer what are the different components of EMG instrument. What are the various factors affecting the EMG signal acquisition? Now, let us address each question one by one. First, to measure the EMG of the muscle, the recording apparatus requires electrodes, amplifier and a recording system. Now, let us discuss about the electrodes. The various electrodes involved in the measurement are the surface electrodes, the insertion electrodes and more advanced 1 and 2 dimensional electrode arrays. Surface electrodes, they are applied superficially. They can be either disc or cup shaped electrode. They are very useful in superficial recording of the muscles, but they also pick up global recordings, which makes them less precise in the recordings. In electrodes, which let us understand the next point of electrode or next type of electrode, that is the insertion electrode. They are either needle type or wire type. They are invasive in nature, but they are very precise. Being precise, they find their use in research. Then the other type of electrode is electrode arrays. They work as a multi-channel EMG. Here many electrodes are arranged into an array and they pick up signals from multiple points and they give spatially sampled image or map of the instantaneous monopolar surface potentials. The extension of information or the extraction of information from these signals give very accurate interpretation of the muscular activity. The proper placement of the electrodes reduces the electrode skin impedance. The electrodes that we are using nowadays comes with already attached gel. When the electrode is applied on the skin, many charge layers are created, which are also called as the Helmholtz layer at the metal gel and the gel skin interface, thereby forming an imaginary or capacitive component of impedance. If we record the impedance between two electrodes separated by gel and between the gelled electrode applied on the skin, we will find that the impedance in the first scenario is lesser than the second. To reduce the impedance, it is also important to prepare the skin surface. The impedance and the effect of skin preparation is a very unique idea and a very unique concept to be understood. In this, the electrode gel skin impedance affects the signal acquisition. The widely varied impedance interferes with the monopolar or bipolar recordings. So, having a good skin preparation is desirable. The best method of skin preparation is rubbing the surface with an abrasive conductive paste and then rinsing, and the other one is washing the surface with soap for 30 seconds and then rinsing. These methods decrease the impedance and improve the signal acquisition. The next component of the EMG machine or the instrument is the amplifier. It is a high gain amplifier in which has a selectivity for frequencies from 10 to 5000 Hertz. The third component of this system is the recording system. In any design of an EMG signal acquisition system, the three principal requirements must be kept in mind. The front end, it interfaces the analog input signal to the analog to the digital converter system. The second component is the voltage reference. It is an electronic device which maintains a constant voltage. The constant voltage is maintained regardless of the temperature changes, the load on the device, power supply variations and the passage of time. The third component is the digital interference or the digital interface. It is the medium through which we interact with the computer. The interaction can be in the form of auditory, visual and functional components like touch or talk. Now, let us discuss the various types of recordings in the electromyography that can be found in a subject. First, the insertional recordings. They are mainly seen while inserting the wire or needle electrode. They should not be considered in the analysis. The presence of these recordings are evidence of the procedure. The next type of recording are the resting muscle recordings. These are the types of recording while the muscles are at rest. It is believed to be the evidence for the activity of the neuromuscular junction. In recent times, it is considered to the basis of the muscle tone. The third point is the muscular activity recordings. The recordings that are associated with the voluntary contraction of the muscles. These EMG signals are seen while the muscle is contracting, and it is the motor unit action potential or MAUP in simple words. At this last part of the module, we will be discussing about the recording of the signals. Now, what are the important factors that affect the recording of the system? One is the movement of the cables. Second, the movement of or the momentarily loss of the skin electrode contact causing a motion artifact. The third point is fluctuation of the charge distribution on the skin and the connecting cables. And the final point is the power line interference. Now, there are two different types of recording system in EMG based on the location of the electrode. One, the monophasic recording. It is mainly done with the monopolar electrode. In monopolar electrode, the active electrode is placed on the muscle and another electrode is maintained at a constant potential, but it is located far from the active electrode, or that is indifferent electrode. A monophasic electrode mainly record the absolute potential because the indifferent electrode is mainly maintained at constant potential. Now, to begin with, how does this happen? Let us suppose the first thing that happens inside the muscle is the muscle fiber is polarized and the wave of depolarization has not reached the segment of the muscle. So, there is no reading in the equipment. Now, the wave of depolarization arrives at the muscle. This polarity is changed now and the excursion is picked up by the electrode. The wave of depolarization now passes away from the muscle and the membrane is repolarized now. The excursion recorded earlier now moved towards the baseline. The action potential has now moved away from the vicinity. Thus, the recording now reaches the baseline, and that is how a monophasic recording takes place. The other important type of recording is the biphasic recording. In EMG, we also use bipolar electrodes. The recordings show the potential difference between the tips of the electrode. Here, two active electrodes are placed side by side at a very close vicinity. The recording is biphasic. Now, let us understand how it is biphasic. Let us assume the shaded area in the nerve impulse. In the initial part of the muscle fiber, it is polarized. The activity that we get is considered the baseline, and both the electrodes are at the same potential. The arrival of the action potential or the wave of depolarization reverses the polarity. The first electrode becomes negative in respect to the second electrode. In EMG, the electrodes are placed extracellularly. So, the first electrode will get an excursion in the reading. Now, the wave of depolarization reaches the second electrode. Here, both the electrodes are in the same polarity. So, the recording comes to the baseline. The first electrode is now undergoing repolarization, which makes it positive in respect to the second electrode, which is still in the depolarized state. This will lead to an excursion in the opposite polarity. The second electrode is repolarized now completely. So, both the electrodes are at the same polarity. Now, the recording comes to the baseline. Thus, a single action potential gives rise to a biphasic recording. Now, we have reached the end of the module. Here, in summary, we will discuss what happened throughout the module. We have understood that the muscle provides the structural support of the body along with the bones. Various activities undertaken by the body is mainly by the activation of motor neuron pool. The motor neuron supplies to various muscle fibers, which forms motor unit. The action potential activating a motor neuron will eventually depolarize the associated muscle fibers, thus generating the motor unit potentials or MUP. In EMG, we record this motor unit potentials and the signals are interpreted by the EMG machines. The recordings depend on various factors like the volume conductor, the skin electrode gel impedance, the distance of the electrodes from the recording surface, etc., etc. The recordings are bipolar or monopolar, which depends on the type of electrode that are used. in predicting the or in interpreting the acute signal, we can diagnose various diseases, and that is what is the beauty of this electromyography. So, now before I end this module, I hope that you have liked the module. Thank you.